and welcome back to Caribbean Online Learning. My name is Keisha Lewis, and in this episode, I will be answering a CSEC exam question on genetics and variation. This is the second in a three-part series. So, let's begin. Now, for this episode, we will be looking at the question from January 2014, question 6, parts A to C. Now, let's look at part A. It says, the differences among individuals are due to variation. Distinguish between continuous and discontinuous variation and give one example of each type of variation. Now, we are being asked to discuss continuous versus discontinuous variation. Let's look at continuous variation first. Continuous variation shows relatively unlimited versions of a characteristic in their phenotype. So this would cover traits such as height that does not have distinct categories. People aren't only 4 versus 5 feet tall. People can be 4.5, 5.8, 6.1 feet, and so on. The graph of distribution is a line graph in the form of a bell curve. It does not have to be a perfect bell curve. Let me show you what I'm talking about. So I'm going to draw a bell curve. Here we have our X and Y axes. One example that we can use here is height. The characteristic, for example, height, is placed on the x-axis. For a distribution curve, the y-axis is usually labeled frequency, with units that can either be numbers of individuals or percentages of the population. And this is our perfect bell curve. Now, not all bell curves are going to look that way. Sometimes we can have one that is skewed to the left, as we can see here, or we can have one that is skewed to the right. We can even have a bell curve that has two peaks. This is usually a special case, such as one species splitting into two over time. All of these are different kinds of bell curves. And now back to where we were before. Some examples of continuous variation are height and skin color. Now let's look at discontinuous variation. This type of variation shows a few, usually 2 to 3, distinct versions of a trait. Many examples are simply the presence or absence of said characteristic, as was stated here. As a result, the graph of distribution is a bar graph. And now we're going to draw a bar graph. As with the line graph, we are using an x and y axis, and instead of a curve, we're now going to draw several bars. Each bar will be representing a distinct point of data. As before, we have frequency on the y-axis, and the example I'll be using is blood groups, which are A, B, AB, and O, each represented by a separate bar. Now, contrast this with the curve that one uses to represent continuous variation. There's no such range of data points here. Discontinuous variation presents very limited options for the characteristic. Other examples are ability or not to roll one's tongue and attached versus unattached earlobe. And that's it for part A. Let's move on to part B. Down syndrome arises from a chromosomal mutation. How does the chromosome number of a person with Down syndrome differ from that of a normal person? Simply put, A person with Down syndrome has one more chromosome than a normal person. This is the result of having a partial or complete extra copy of chromosome 21. And that's it for Part B. Part C. Sex-linked inheritance occurs when the genes are on the sex chromosomes. In most cases, the gene is on the X chromosome and not on the Y chromosome. Those of you that looked at episode 1 of this series would have come across the phenomenon there as well. And just like episode 1 question, we are being asked to use a Punnett square to determine the possible genotypes of the offspring of a cross between a female carrier and a normal male. Unlike episode 1, you have to complete the Punnett square and write out the genotypic plus phenotypic ratios of the offspring. So that's what we're going to do now. Before I complete the Punnett square, let's determine the alleles that will be present in the gametes of the parents. Let's start with the genotypes of the male and female parents. The genotype of the female is X superscript capital H, X superscript common H. A cell with this combination would produce gametes with the following proportions. So the gametes produced by the female would be 50% X capital H and 50% X common H. The genotype of the male is X superscript capital H, Y with no corresponding allele. This is what makes the condition sex-linked. The gene in question is carried only on the X chromosome. This makes males more vulnerable to having the condition. The gametes produced by the male would be 50% X capital H and 50% Y with no corresponding allele. Now that we know what our gametes are, we can now complete the Punnett square. And here we have our Punnett square. I will just add a line here to divide the male section from the female section. The male gametes will be lined up horizontally and the female gametes will be lined up vertically. The gametes from the male parent are written in the header row and the gametes from the female parent are written in the header column. And now I'll write the possible combinations. This section will be a combination of this gamete and this gamete. And this section will be combining this and this. And this section will be a combination of this gamete and this gamete. Note that the X is written first, and so on. And that's your completed Punnett square. Now, from the new combinations produced, we can tell that this offspring would be normal. This male offspring would also be normal due to the presence of the normal allele. The other female offspring would be a carrier. She has the disease-causing allele, but the presence of the dominant normal allele would keep her from developing the disease. And the other male offspring would have the disease, having just one allele, which is the disease-causing one. There's no other allele present to suppress its expression. And now, as required by the question, I'm going to write out the genotypic ratio and phenotypic ratio of the offspring. So first I will write the offspring genotypic ratio. And here we have a 1:1:1:1 ratio, which translates to a 25% probability for each combination. So how does this translate into phenotype? Once again, we have a 1:1:1:1:1 ratio, which translates to a 25% probability for either phenotype. Take note that only the male offspring shows the probability of developing the condition of red-green color blindness. This confirms the sex-linked aspect of this condition. And that brings us to the end of this question. I hope this video has been of help to you. If so, please subscribe and click the notification bell. More videos are coming, including the third and final episode in this series of past paper questions on genetics and variation. Videos for CAPE Biology are also on their way. So subscribe today and see you soon.